Okay, now we're ready. Okay, so to begin this section has to do with how do you get a protein that the cell has made to its correct location. And a lot of times that location is in a particular organelle, or it might be going to out to extracellular space outside the cell. And when you have organelles in a cell, that adds a lot of complication to getting proteins where they need to go. So why is it useful for cells to have organelles? What's the advantage of having organelles? Yeah, they each have their own job and they're specialized in function. So organelles conceptually are separate spaces with separate conditions. And they are doing specialized tasks. And thinking about the benefit of organelles, it's useful to think about how do eukaryotes function compared to prokaryotes, which do not have organelles and they only have one compartment, generally. So organelles allow greater complexity of function. It's kind of like having a house with multiple rooms, and some rooms are specialized to do certain jobs, and not every single thing that takes place has to be in the same room. So organelles are good in that way, but having organelles means you have to get all the different proteins sent to their correct location. So it adds complexity of function, but it's also a little bit complicated for the cell to get proteins to their correct location. In a real cell, kind of like we saw in that video, there are a lot of organelles in a fairly small space, and the cytoplasm is much more crowded than maybe you might expect. So in the average cell, the organelles take up about half of the space in the cytoplasm. And the word cytoplasm actually is referring to all the stuff that is inside the plasma membrane. So it's both the liquid component, which is cytosol, or the stuff in between. So when you look at a picture like this, the stuff in the gray is the cytosol, the liquid. And when you add together the cytosol plus the organelles and plus the cytoskeleton and plus the ribosomes, all of that together is cytoplasm. The cytosol also contains all of the dissolved small molecules in the cell. Or we would say the soluble components. And that's going to include sugars, your organic chemicals, organic chemicals, ions, ATP, all kinds of small molecules. And anything that is soluble in the cytoplasm is going to be able to diffuse place to place. So if we have lots of ATP being produced in the mitochondria, and then they get sent outside the mitochondria, those ATP are able to diffuse all through the cytoplasm and be used just anywhere. So very important concept. Anything that is dissolved in the cytoplasm is going to diffuse all over the cell unless something grabs it and pumps it, or moves it on purpose. It will diffuse everywhere in the cell. So this table, this exciting table, is showing what are all of the different compartments of a cell. What are all of the enclosed spaces or the separate rooms or the organelles that a cell can have? And I think you're really pretty familiar with these, but there's one that you don't maybe learn, at least I didn't when I was learning about organelles, which is endosomes. This word endosome. And an endosome is a membrane enclosed temporary organelle. And it connects conceptually with some other compartment. I'm going to look now and see how they define endosome. Sorting of endocytosed material. Yeah, kind of. Let me add on to what I'm writing about endosomes. Endosome is a holding tank for materials that are on their way between certain compartments. And this is where they might be heading between. So materials that are heading between extracellular space lysosomes and the ER/Golgi. Endosomes are kind of an in-between holding spot, or like a storage room in your house for materials that are heading between those compartments. And notice on here, they are calling the cytosol a compartment, and it is. We don't think of it as an organelle, but it is a separate location that exists in a cell. So this table kind of is a list of organelles, or it's a list of compartments, and you can also think of this as a list of destinations that proteins need to find. So if there's any protein in the cell needs to get to one of these locations to do its job. So getting the proteins that are made by the cell to their correct location is the topic of, I think, today and Thursday's lecture. How do proteins get to these different compartments and get sent to the correct place where they're supposed to do their job?
How come you wrote those extracellular lysosomes on the
These are the actually add on extracellular to this list of compartments. Extracellular space is not technically part of a cell, but it's a destination where proteins might need to go. And these compartments together represent kind of a connected set of destinations called endomembrane system. And there's a slide coming later that shows what are the parts of the endomembrane system. Yeah, good question. Extracellular is a destination. It is a compartment, even if it's not technically inside the cell, because anything a cell makes and it needs to send outside itself, those proteins have to go to that extracellular area, let's say.
So that was for extracellular? Is that what you said? What was that?
This is a list of destinations.
Okay.
And it would be helpful to add onto this list extracellular as a destination. Good questions. This section, it tends to be one that it doesn't come up in the previous classes that you've had. Usually. So newer stuff. So what's coming next is we're kind of going to look at some different compartments. And the first one that we're going to look at is the nuclear compartment. So focus most on this picture here. This could also be a modern cell. So inside the nucleus is a space of its own, and there's a membrane around it, which is double layered. Kind of like a mitochondrial membranes are double layered. There's an inner section and there's an outer section. And nuclear membranes tend to be folded up in such a way that they are continuous with the endoplasmic reticulum. Is that a new fact or an old fact? Like, did you ever learn that before? I swear I did not. It's good that you did, if you did, but nuclear membranes are continuous with the endoplasmic reticular membranes. It's double layered, and there are pores within the nuclear membrane to let things get in and out. This slide we're going to skip over and go here. So this cartoon is showing, okay, so the cell is making proteins in the cytosol because that's where the ribosomes are. So all proteins start out being produced in the cytosol, but this picture is color coding them to show where are they supposed to end up to do their job. So some of them would need to end up in a peroxisome because that's where their job is waiting for them. Some of them would need to go into the nucleus, some into a chloroplast, some into mitochondria, and then some of them into endomembrane system. All proteins start out getting made in the cytoplasm, but getting to the correct location requires this whole system of the cell to identify and tag proteins for their destination. That's how they're going to find their place. And here's how proteins find their place. They have encoded in the sequence for their genes something called a signal sequence. So this would be in the DNA, and then when the protein gets produced, there are amino acids produced in that protein that serve as a signal sequence, which is like a tag. The signal sequence is like a tag in or on a protein. gives a little 3D shape. And that little 3D shape that's part of the protein sequence of this protein is going to allow transport to the correct compartment. That's how proteins know where to go. They have a part of them that is built into them that has a little shape of certain amino acids. They're going to fold up into a 3D overall shape. And that will tell the protein or let it get dragged to its correct location. The point of this chart is not to memorize the what the signal sequences are, but to show the variety of what they can be. So some of them are N-terminal. We can have N-terminal tags, and those would come out of the ribosome and fold first as the first part of the protein that's made. So an example of that is when you see on here signal sequence and it says plus H3N, that's the amino or the N-terminus of the protein, and this would be constructed at the very beginning of translation, the N-terminal tag. And when you are looking at these sequences, the amino acids with certain colors are more important than the other ones. So if a protein has this type of N-terminal tag, it is going to make that protein go into the ER, enter into the ER, and enter that pathway. There are also C-terminal tags. They're the last thing to be produced during translation. Come out last. They're the very ending thing. And those are denoted on here when you see COO minus. That's the carboxy terminus, C-terminal end of the protein. And if a protein has this particular type of tag, it tells the protein to stay in the ER. It's a signal to stay inside the ER and not go elsewhere.
C-terminus?
That particular C-terminal tag. There are other, that's not the only C-terminal tag that exists, but and then these two examples that we see down here are internal. Internal tags are in the middle of the protein primary sequence. So produced in the middle of translation. And these type of tags tend to kind of like stick out like a bump on the surface of the protein. And depending on what the tag is, it might cause the protein to go to the nucleus or to go to a peroxisome. These are not all of the tags that exist. It's just examples. Just examples. The other note to make about this is if a protein has no tag at all, no signal sequence, it is going to take the default, it's going to go to the default location, which is the cytoplasm. And if proteins that have no special signal sequence will go to will just stay in the cytoplasm. the lack of a tag means you don't need to go anywhere special. You can just stay in the cytoplasm and do your function there. That's where we're going to have our break today. And this star note, I'm going to come back to that when we get to a later slide. It'll make more sense when we get to a later slide. We'll have our break right now. Did you all have a quiz today? Okay. I thought I might need the Kleenex today.
Yeah.
There was a student in the nursing micro class that was like cutting stuff yesterday. But I had never, she doesn't usually. So I thought, what just changed?
She's copying me.
Maybe, I don't know if you know each other. Katie, do you know Katie Biggs?
I've heard that name.
Yeah. She has 2 classes with me at once. So she's doing great. So lots of tags exist. We're going to highlight two different pathways to go into two different locations. And the first one is, how does a protein get into the nucleus if that's its destination? Nuclear tag is internal, and it's kind of going to be sticking out of the protein. Like if I had constantly like a fanny pack that I wore and it was always attached to me, it just kind of stuck out of my middle. That's what a nuclear import tag kind of acts like. Oops, it's the wrong direction. So proteins that need to go into the nucleus to do their job, they're going to have a nuclear tag, and they are going to move through these pores in the nuclear membrane. So they would be produced in the cytoplasm, and they need to travel through a pore to get into the nucleus where they're going to do their function. Something I want to highlight on this slide is something called nuclear lamina. This is going to come up toward the end of the semester, but this is the first place we have a picture of that. So nuclear lamina is a set of structural proteins that kind of hold up or support the nuclear membrane. And I think about these as like tent poles. So you've got this large membrane system surrounding the nucleus and surrounding the DNA that's in the nucleus. And there are some support proteins that kind of give a little bit of strength to that membrane. And you have heard of these before when you learn about the stages of mitosis. And when you learn about prophase, that during prophase, I now quote generations of textbooks, the nuclear membrane breaks down and blah, 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 blah. That's going to be this lamin support system coming apart. And that's what allows the nuclear membrane to break down, and then the chromosomes condense and all of that. So this is the first place we've got a picture of that, so I wanted to highlight it. What we're going to look at next is the details of, okay, how does the protein that needs to go into the nucleus get from cytoplasm through the pore into the nucleus? So we're going to look at nuclear pore complex. And there's a part of this picture that is, oh yeah, that looks better. Easier to see with lights off. Nuclear pores are not just open holes that let anything go in and out. They have all of this other structure to them that looks kind of weird, but it has a purpose. So what we're looking at here is this would be inside the nucleus, and here's the nuclear lamina in green. And here's a nuclear pore, and that pore is lined with proteins that have a very weird structure together. So there are these long extensions that poke out into the cytosol called cytosolic fibrils, pointing out that way. There is some unstructured loop in the middle of the pore that's kind of acting like a filter or something. This would be unstructured loop. And then on the inside section, inside the nucleus, there's this arrangement called nuclear basket, which is nuclear fibril connecting to this binding structure on the inside. The segments of the nuclear pore complex, you don't need to really know, but that's what they're called. And you can see parts of them in sophisticated electron micrographs. So this is looking cross section. This is looking down on the outside of the nuclear membrane, and you can see the ring structure of these nuclear pore complex, which is cool. And this nuclear pore complex is very involved in controlling what does and does not go in and out of the nucleus. So this picture is the same thing again. It's not really adding much. Let's go to this picture. Okay, so now we've got kind of more detail of what the nuclear pore complex looks like, and this always looks like a donut with a cherry on top or something. So delicious. It's a nuclear protein that needs to be doing its job inside the nucleus, and that little red bump is its nuclear localization signal, or its fanny pack, or its dragging tag that's going to help it get into the nucleus. So the way that a protein actually gets into the nucleus is it first has to bind with this other protein that's floating around in the cytoplasm called nuclear transport receptor. Nuclear transport receptor. And a lot of times we think about a receptor as something that is stuck in a membrane and can't move, and it's waiting for something to come and bind it. This receptor can float around in the cytoplasm. And the nuclear transport receptor, its binding partner, is looking for the nuclear localization tag. It is looking for this particular set of amino acids that fold up into a little bumpy shape that stick out of a protein. That's the binding partner of nuclear transport receptor. So these two bind each other well. And when they come close together, they will bind by the four forces. Non-covalently. So they would have a binding attraction to each other. And the combo of the nuclear transport receptor plus the nuclear localization tag is going to be a new combo shape. So when those two bind together, they form a new overall shape, and that combo shape is going to bind to the cytosolic fibrils. Let's call it fibrils on here. Nuclear transport receptor by itself does not bind well to fibrils, and nuclear localization signal by itself does not bind well to fibrils, but the combo of them together does bind well to these fibrils. To me, this nuclear pore complex always looks like a frisbee golf basket thing. Where you've got, if you could kind of get the frisbee into this section, it kind of will make its way downward. That's kind of a similar concept with, if you got this combo together, they can interact with the fibrils and then eventually move on down through the unstructured loop. This part scientists understand pretty well. This part, scientists understand pretty well. This part is still a question mark. Like, once it's up here, how does it move down? That's still a pretty active research area. But it does go through the pore, kind of still in a mysterious way, and ends up delivering the nuclear protein to the inside of the nucleus. So what we're going to look at next is, okay, once these two are in here, how do they come apart? Because they are definitely binding well out here in the cytoplasm. After delivery, how do these two come apart? And that involves a new protein called RAN GTPase. I don't have a picture of RAN GTPase. I need to draw that in. So once this combo is inside the nucleus, the combo of the nuclear transport receptor and the nuclear localization signal are going to get forcefully separated. They don't just dissociate, they don't just drift apart, they have to be forcefully separated by this enzyme called RAN GTPase. And to do the ripping apart, RAN GTPase has to spin the GTP kind of in a similar way as spending an ATP. So it requires energy for this enzyme RAN GTPase to get these two apart and separate them. And then what's going to happen? So if you've got this picture, you could write right here the word RAN GTPase. And then the combo of the protein that's now made it to the nucleus and the nuclear transport receptor, they're going to go their separate ways. This one is going to go ahead and do its job in the nucleus. It's got to its place. It's delivered to the location. It's going to do its function. Nuclear transport receptor will then go back out nuclear pore complex and go to the cytoplasm, and it can pick up a new passenger. This nuclear transport receptor is basically like a shuttle. It just makes a constant loop of pick up in the cytoplasm, drop off in the nucleus, go in and out of nuclear pore complexes all day long.
You said pick up in the cytoplasm and drop off there.
In the nucleus. That's its only job so far. Pick up anything out here that's got a nuclear localization sequence that you can bind to. Drag it into the nucleus. Have it ripped away from you by RAN GTPase. Go back out and pick up some more. So that system, that's how proteins get into the nucleus. We're going to skip over import into mitochondria and chloroplast. Getting proteins into the mitochondria and the chloroplast is pretty complicated because you've got to get across two separate membranes, and there is not a hole that crosses through both of them like the nucleus. Interesting, but we're going to skip over this. If you ever need it, you can come back to these slides and look. And the second system that we're going to look at of protein targeting is getting proteins into the endoplasmic reticulum. How do proteins get into endoplasmic reticulum? There are a lot of proteins that need to go into the endoplasmic reticulum for a variety of reasons. Have you learned before, like, why would a protein ever go there in the first place? What could be some reasons it needs to be in the endoplasmic reticulum? What is the job of the endoplasmic reticulum?
They create lipids.
I think it does. So maybe a protein is involved with lipid creation.
I was gonna say it holds ribosomes.
It does hold ribosomes, kind of for the purpose of getting proteins sent into the ER.
The vesicles that are sent out.
Mm-hmm.
Of the
Eventually, vesicles can come out of this system. Moses, it's in the 7th grade. They're starting a unit on organelles. It seems too early, but anyway. And I'm curious to ask him like what they learned about what the endoplasmic reticulum does, because I think that they really have no conceptual understanding of what a protein is. Or anyway, I don't think they just started this week, so I don't think they've gotten to ER yet. But what Liz said about the vesicle, that's underlying something really important about the ER, which is this. Any protein that eventually needs to go to these locations would have to go to the ER first. So if it's going extracellular space, that might be because the protein is going to be secreted outside the cell. If the protein needs to be in the plasma membrane, or in the cell membrane, it would need to go into the ER first. If the protein is going to go into ER or Golgi lumen, so the space inside them, or one of those membranes, it would need to go to the ER first. If it needs to end up in a lysosome, it needs to go to the ER first. And if it needs to, at some point, go into a vesicle, it needs to go into the ER first. And normally, the vesicles are transporting between these locations. In any of those situations, the protein needs to get into the ER to get started on that journey, if it's going to any of those places. I did not, until I understood that, I didn't really get the point of the ER. Like, I memorized before, like it's involved with targeting proteins and sorting them for blah, blah, blah. The ER is involved with getting proteins where they need to go in the cell to do their job. And it's the first destination for so many proteins that are going any of these places. So to get into the ER in the first place, a ribosome has to put you into the ER. And the ribosome that's putting you as a protein into the ER is going to be associated or stuck with the membrane of the ER. And that's what you've learned is rough ER. So I remember memorizing that. Rough ER has ribosomes on it. Okay. The reason the ribosomes are there is to start building the proteins into the ER so that they can go to their destination. So much on this slide. How does this seem so far? So now we look at, okay, how does the ribosome get the protein into the ER? So what we have to imagine first is that, let's go back and look at that table that has the different targeting sequences. Okay, so that's why the star is here actually. The star is supposed to be next to this, just to note that any of these type of proteins would have to go to the ER first. And what the star really means is that all these would need the N-terminal ER signal sequence. Lots and lots of proteins have N-terminal ER signal sequence because it's the starting destination to go all of those other places. If you are a protein that needs to stay and do your work in the ER lumen, you would have this to get you there. And then you would have this to make you stay in there. So proteins can have more than one tag at once if they need to get to a location and stay at the location. They could have two separate tags. So if you are a protein and you have that N-terminal sequence tag, this is how your book draws it, as this brown blob. Or, I'm sorry, they're drawing it as the red line. I need the colors on this a little bit better. Okay, so your protein, your destination is one of these places. And as the ribosome is translating the messenger RNA that encodes you, the first part of the protein that comes out is that N-terminal ER signal sequence. The very first thing that comes out, and that's going to fold into a 3D shape that combined to this protein, the brown double bump protein called signal recognition particle. And we're going to just abbreviate that SRP. So this is SRP. This red segment here is the signal sequence that's a part of the protein that's supposed to get into the ER. So now that we've seen the colors, with the lights back on, so we can see the board writing. So this signal recognition particle is conceptually similar to the nuclear transport receptor. It floats around in the cytoplasm. And its binding partner is the ER signal sequence. And you would have multiple of these SRPs floating around in the cytoplasm, just kind of waiting around to bind the binding partner through the four forces whenever the binding partner appears. And when those two get together, so if you have ER signal sequence, we'll call this ERSS. So SRP together plus ER signal sequence. When those are together as a combo, they are going to bind this protein called SRP receptor. And that's at the ER membrane. SRP receptor does not bind just SRP, and it does not bind just the ER signal sequence. It binds the combo of them together. And that binding event of those binding together ends up dragging the ribosome to the ER. So the ribosome, as it is still working, as it is still translating, gets dragged over to the ER by this combo binding event. When I first memorized that rough ER has ribosomes on it, I imagined that the ribosomes like just live there. They're always there. They're not always there. Let's look back at that. So any ribosome that's on the ER membrane is there just for a little while as it's translating, and they actually come and go off of the ER membrane. Did not realize that. So I thought, okay, if you're a ER ribosome, that's just like, that's your life. You're stuck there all the time. No, you get dragged there as you are translating a protein that has an ER signal sequence on it. Something interesting about this, when SRP is bound to the signal sequence, it kind of slows down the ribosome. So this picture, this is my favorite cartoon picture in the cell book because of the stage in my life where I first saw this. So when Han and I came here, we had Noah was 6 and Moses was less than 2 and Joseph was 5 months. Is that right? Moses was 2, exactly 2. And the first year that I taught cell, it used to be in the spring. And when I was teaching this class in January, right after we came back from Christmas break, the Moses daycare teacher said, you know, you really need to potty train him. And he was 2.5, and based on experience with Noah, 3 for boys is way easier than 2.5. And I was so mad because we had just had Christmas break, and I thought, why couldn't you tell me that before I was home with them for 3 weeks? That would have been a good time. I can't do this now. And then so I still started trying to potty train him during the semester, which is hard. And I remember coming to this picture, and there's something coming out, and there's this turd looking thing. And like, here's where the hole where it's supposed to go. And I just like learning that this slows down the translation. It reminded me of like, oh my gosh, he's about to poop. And you pick him up, you're like running to the toilet, you're trying to get your butt over the hole so that they can put the item in the correct place where it's supposed to go. And I just thought, this is the best thing I've ever seen. This is like my life right now. Slow down, slow down, please, your production, until we can get to the location where you can put it through the hole. So the hole that this protein is supposed to go through to get into the ER is called translocation channel. And the translocation channel is able to take this protein, amino acid chain, as it's being produced and thread it through that hole. I think that's, yeah, this slide. It's the same picture, different words. Okay, so kind of looking at this section now. So SRP receptor, once it's bound to this combo, is able to kind of hand off this polypeptide chain that the ribosome is making and move it over into this translocation channel. So you can kind of see right here, it's not continuous. It's kind of got a little sideways entry into it. So what happens is the ER signal sequence is going to be put into the membrane and it's going to stay there. So it's going to stays with the ER membrane. And the rest of the protein is going to be translated through that translocation channel into the ER lumen. So the ribosome is kind of going to stay here while it's translating, and it's by being here, it's making the ER rough. And it keeps on translating this messenger RNA into protein, but the protein, as it's being made, is coming through this channel. So moving from the cytoplasm into the ER lumen. But this segment of this protein is going to stay stuck in the membrane. And it will kind of hold this protein together with the membrane unless the cell decides it needs to cut that off. SRP, once it has gotten the ribosome to the SRP receptor and kind of handed off this situation to the translocation channel, it comes off and floats around in the cytoplasm again, looking for another ER signal sequence to bind. And then the protein has, gotten near its location, and it can keep being translated. And what happens to it next depends on what kind of protein is this. So if a protein is supposed to be soluble, what they're calling here a mature soluble protein, that indicates that that protein will not be a membrane protein. So if the protein's ultimate job is to float around in a liquid and not be together with a membrane, that's a soluble protein. And if this protein that's being translated into the ER lumen is supposed to do a soluble type of job, it will go ahead and fold up into its 3D shape. And then there are some enzymes that cut off that signal sequence, and it just kind of stays there. And this protein has now been delivered, and it's in the ER lumen. It's folded up into its 3D shape. And it can then be delivered to the correct location to do its soluble job. What if the protein is not supposed to do a soluble job, but it's supposed to be with a membrane? It has a little bit more complicated situation. So if this protein is supposed to be doing a job where it's supposed to go through a membrane and be membrane integral. Any kind of membrane protein. So any of those membranes that we talked about, like it could be the cell membrane, vesicle membrane, ER membrane, Golgi membrane. It's going to have the ER signal sequence. To get it to the ER, it's going to be translated into the lumen, but it can have a segment inside it called a stop transfer sequence. And this stop transfer sequence is going to become the membrane inserted segment of the finished protein. So over here we're seeing the finished protein, and this type of protein is supposed to have sections of protein on either side of the membrane. So we've got this segment is built into the ER lumen, and then we've got this transmembrane section that holds it in the membrane, and then this segment is supposed to stick out the other side. They've got on here, this is the cytosol, this is the ER lumen. If you need a protein to be built into a membrane, this is how it stays in the membrane. So it basically, when this reaches the translocation channel, it signals the ribosome to just translate the rest of this on this side. And the protein can be built into the membrane in this way. It's a good place to stop because it's time to stop. So next time we're going to look at, okay, if this protein is supposed to go other places, how does it get from the ER to its next location? That's the Thursday topic. Thursday.